This is our last module in the Jepson mini workshop, Biodiversity Challenges, California Floristics, Taxonomy, and Phylogenetics. And here I want to circle back to the first module in thinking about the continued discovery with regard to really fine scale discoveries down at the level that would correspond to species or even finer scale taxa in this module called Cryptic Diversity and Phylogeny-based Taxonomy. So the term cryptic diversity gets tossed around a lot, and I don't particularly like it, even though I actually have been involved in promoting this idea, or at least the importance of this concept. But the bottom line is that these 3 points made below here are the important thing, that what we generally call cryptic diversity are distinct evolutionary lineages warranting taxonomic recognition that are difficult or impossible to distinguish morphologically, but that may not be ecologically interchangeable, in which case they represent irreplaceable diversity that's important to consider in conservation planning. For example, these different lineages that are hard to tell apart may have really distinct climatic adaptations or adaptations to soils. They may be important groups that are evolutionarily divergent. If we know they're evolutionarily divergent, then that's not even a question, but that represent situations where ecological change is happening more rapidly than obvious morphological change, which is something that we know already is common in nature. So definitely cryptic diversity is not the same thing, the botanical equivalent of cryptozoology, which if you don't know what that is, you can look it up online. I probably won't get into it, but it has to do with Sasquatch and the Loch Ness Monster and the like. So definitely don't equate the concepts of cryptic diversity and cryptozoology or cryptobotany. So I just want to show you a few case studies from California from previously well-studied flowering plant groups in different plant families that are cases where we have cryptic or semi-cryptic lineages that are not each other's closest relatives. So based on any criterion, or monophyly criterion would have to be treated as distinct species unless we do a lot of lumping. And where there's also direct or indirect evidence of ecological differences between the cryptic species. So they're very likely or definitely not ecologically interchangeable. And this, of course, has a lot of importance in terms of conservation and making sure that our taxonomy reflects the diversity of life that's really out there that represents ancient and irreplaceable diversity. So the case that I always point to first are the common goldfields, which David Keck, that I mentioned earlier as part of the Clausen, Keck, and Heise biosystematic team, suggested was the most abundant native composite in the state. And as you're probably aware, common goldfields forms extensive wildflower fields, are beautiful annual members of the sunflower family. And it's long been noted that within what's traditionally called common goldfields, Lasthenia californica in the broad sense, that there was a lot of biochemical and floral variation within it. Floral variation being variation in the pappus, which are homologous to sepals at the top of the fruits here. And here you can just see some of the variation in disc flowers in this selection of florets here. So this understanding of biochemical and morphological variation goes back to work by Bruce Bohm and Bob Ornduff, who I've been fortunate to overlap with in my career. And one of my early grad students at Berkeley, Raymond Chan, took on a study of Lasthenia while Bob Ornduff was still living, which was really wonderful, and did an extensive sampling throughout the genus and within Lasthenia californica in the broad sense, and was able to show from both chloroplast DNA and nuclear DNA phylogenies that Lasthenia californica it's not a natural group in the way it had been circumscribed, that the core are typical Lasthenia californica that has narrow pappus elements that can often be tawny or brownish, is more closely related to Lasthenia macrantha, the perennial species at the time recognized within Lasthenia. And californica was less closely related to this outlying lineage of Lasthenia californica that had traditionally long ago had the name applied to it of Lasthenia gracilis, which has a broader base to the pappus elements and is really glistening white rather than tawny. So there were morphological markers for recognizing these two as different species and treating macrantha as a subspecies of californica. The problem was that there are populations of both of these species in their new circumscription that don't have a pappus at all. And if the pappus is lacking, you're really on your own. I mean, there's no really good way to tell them apart. And yet they are evolutionarily distinct. And not only that, they show some degree of geographic separation, but they overlap extensively. And it turns out to be even greater than shown here based on later sampling. But they are ecologically distinct. And we know a lot about this based in part on the earlier work of Bruce Bohm, but also by the work of his graduate student, now professor at Cal Poly San Luis Obispo, Nishi Rajakaruna, and Jen Yost, who is professor also at the same university, Cal Poly SLO, but was a student of Kathleen Kay at UC Santa Cruz, who was a postdoc with me at Berkeley. And they basically both have done some really nice work in the common goldfields. And in collaboration with my lab, Nishi, using the biochemical evidence that Bruce Bohm had generated earlier, we came to realize that where the two species overlap and grow together, they occur in different soils. So serpentine versus non-serpentine, or I should say extreme serpentine that has a really low calcium magnesium ratio versus less extreme serpentine with a more equitable ratio of calcium and magnesium, or saline versus non-saline. And previously, two biochemically and ecologically distinct races were recognized in these situations, like at Jasper Ridge, where we now know these biochemical races are different species. But the crazy thing is that if you go to different parts of their range and find them together, the different species include both races. So they always have a reciprocal adaptation, but sometimes to a different, to the more extreme situation or to the less extreme situation, depending on the site. So there's been some parallel evolution going on there as well. And Jen Yost showed through reciprocal transplant studies and a lot of intensive work in these contact areas that there is definitely differential adaptation where they grow together. So this is a case of cryptic species, truly cryptic without their pappus, that are ecologically differentiated and evolutionarily divergent. In the broom rapes, again, that I talked about earlier with regard to higher level classification, Adam Schneider and colleagues like Alison Caldwell have worked on the finer scale relationships within the New World broom rapes as well. And here you can see the results of work that Adam spearheaded, part of his dissertation in Aphyllon, the New World X or Orobanches. And you can see a lot of phylogenetic structure here that's not represented taxonomically. And you can see that the species in this one part of the New World broom rapes, Aphyllon uniflorum and Aphyllon fasciculatum, include some really divergent clades. So in the case of Aphyllon uniflorum, there are these two really well-supported clades here that are most readily distinguishable based on their different host plants. And you can see in this clade, there's even finer clade structure with different hosts very different host plants. The orobanches are root parasites. Orobanchaceae are root parasites that have haustoria that attach to the roots of their host plants, and they're completely dependent on their host plants for nutrients. They are holoparasites, non-photosynthetic. And it was thought that, for example, these two species, Uniflorum and fasciculatum, had really broad host range, but it turns out instead that they've just undergone much more rapid ecological shifts to different hosts than the rate of morphological change. And let's face it, there's not a lot of morphology there anyways. They're highly reduced holoparasites. And so now this clade of Aphyllon uniflorum is now called Aphyllon purpureum, which has included with it some additional lineages that have host specificity that could be recognized as taxa as well. And if we look at the Aphyllon clades, what we can see is that they're very widespread geographically oftentimes. And what is actually, so there doesn't appear to have been allopatric speciation going on here, geographic speciation, but instead we see a stronger association with hosts, and it's really shifts between hosts that appear to have driven the diversification of the group. I want to focus in now on a segregate of Aphyllon fasciculatum in the broad earlier sense that has been recently recognized by Allison Caldwell and Adam Schneider and colleague, that is called Aphyllon epigalium and is strictly endemic to the genus Galium as a host plant. And you can see it here, and it's primarily Californian distribution. There are two subspecies recognized that are morphologically differentiated if you look carefully at them in terms of floral morphology, for example. And there we might actually have a case of geographic speciation, but probably also involving climatic factors and possibly other habitat considerations given the difference in climate in northern and southern California here. But I wanted to bring up this particular example in part because this was this particular species was described in 2018, but 100 years earlier in 1918, Willisleen Jepson himself put an annotation in his own hand here on this specimen of a plant that corresponds to the new species, Aphyllon epigalium, where he pointed out that it has some features that don't quite fit for either uniflora or fasciculata. And moreover, the flowers are yellow in color, which is not right. So this just goes to show you what I was mentioning earlier, that the herbaria are major reservoirs for discovery of new biodiversity. And this was hiding away here with a hint even from Jepson himself. Also, for example, in the Navarretias and the phlox family, we have really rapid diversification that's happened in this clade. Here you can see 6 species that were not long ago treated as one species, but differ in their ecology and their ploidy even. You can see different chromosome, this 4N versus 2N ploidy. The easiest way to tell these apart is by the names on the photographs here. But they don't come with those names attached to them in nature, unfortunately. And you really have to look closely at the flowers and the vegetative features to distinguish them. But they're very good clades, very good lineages that are ecologically distinct. These two most recently described by Lee Johnson are each serpentine endemics on either side of the Central Valley. So there's a lot of important diversity here. Looking at fresh material, there's really no substitute for that. Sometimes when we talk about cryptic diversity, what we really mean is once they're pressed and dried, they're very cryptic. And you see them in the flesh alive, they're less so. And finally, with the monkey flowers, again, going back to them, Naomi Fraga has done some marvelous work with extensive sampling within what had been previously regarded as one species, and found that here in 2 different cases, and there are others as well, that what had been thought to be one species of monkey flower turns out to be multiple, never before describe species or species that had been recognized earlier and placed in synonymy. So the phylogenetic data, the molecular data, has really been helpful in teasing out these lineages that are often quite distinctive ecologically. But once you press them, it's often hard to see these differences, for example, that are so striking in these photographs. And I'll just finish this part of the workshop, this last part, by going back to the tar weeds again one last time. One of the rarest species I've had the pleasure to work with, or what I thought was a species initially, was Lagophylla dichotoma. And it had occurrences along the east edge of the Sacramento Valley and even out into the Sacramento Valley historically. But those have all contracted down now to 2 localities in the southern Sierra foothills. And then there were a bunch of localities in the South Inner Coast Ranges down here. Well, it only took one trip out to these 2 sites, or 2 populations in these 2 sites, to realize these plants are not close, or not very similar morphologically. When you press them and dry them, they look amazingly similar, but once you see them in the flesh, the differences stand out in ways you can see in the pressed specimens. Turns out that these western populations in the South Inner Coast Ranges are more closely related to the widespread Lagophylla ramosissima than they are to Lagophylla dichotoma on the other side of the valley. So I now recognize another species for these, a new species for Lagophylla. What had been dichotoma on the west side of the valley is Lagophylla diabolensis, being in the Diablo range here. And this reduces the circumscription of Lagophylla dichotoma down to 2 extant populations. So it is exceedingly rare and really a scary prospect for the future. I mean, it was even rare in the 30s when David Keck wrote about it as being one of the very rare tar weeds. But one of the things we also noted that I want to bring up to close is that not only are these populations really obviously morphologically distinct when you see them in the flesh, but another thing when you see them alive is this particular species doesn't have a flower moth associated with it, whereas this species does. There's a flower moth that is associated with this species. We immediately found walking around in these populations. No sign of them out here. These flower moths are in the are heliophynes. They're in the case of Lagophylla, they're moths in the genus Microhelia. And there are a large number of other tarweed moths that Terry Sears, Lepidopterus, has recently discovered and recognized that amplify the number of tarweed moths that are known and that are completely dependent on tarweeds feeding on their foliage before they pupate and are restricted to particular clades. So these different clades of tarweed moths are restricted to particular clades of tarweeds and depend on them. They represent a much greater diversity than we thought. So there was cryptic diversity of tarweed moths, cryptic diversity of tarweeds. Their wing patterns do differ somewhat if you look closely at them when they're adults. They're beautiful moths that nectar on a wide variety of different flowering plants. And Terry even found one species of flower moth that had never been described that had a really distinctive larva, and that was a specialist on a subset of the lineages in Calicodenia multiglandulosa, tarweed species. So this just kind of brings home the final conclusions I want to make, that California plant taxonomy still has a frontier at home, and that species or subspecies or varieties should not be treated as ecologically or evolutionarily uniform in the absence of good data to that extent. There could be cryptic diversity lurking within those groups, and the loss of that cryptic diversity is extinction, regardless of whether it's named or not. And it could take with it associated organisms like the tarweed moths, all of these being irreplaceable parts of food webs that are just now becoming understood with regard to the arthropod food webs of tarweeds, for example, with the work of Billy Krimmel and Ian Pierce and others, Eric Lopresti, in the case of that particular situation. And the lack of such detailed data on lineages within most species really warrants and makes a great argument for conserving species or other taxa across their ecological and geographic ranges. And importantly, very cautious mitigation in terms of which is critical when considering irreversible actions like genetic augmentation, moving seeds from a population that's going to be destroyed into a population far away, which might represent a cryptic lineage that'll be swamped out by that germplasm. So, there's a potential to do more harm than good with mitigation. And we need to be careful about such things as genetic augmentation and assisted migration and the like without better data. Okay, so that takes us to the end, and I definitely open it up to questions or comments.